How does processed dog food affect reactivity and aggression in dogs? Recognizing the escalation of arousal into aggression brought on by the fear reflex of fight or flight, these investigations look at a connection in diet being a contributing factor in a constant state of being, making reactivity more likely. So what specifically relaxes or heightens the state of mind? To find the answer to our question, we need to take a closer look at the brain, specifically neurotransmitters dopamine and serotonin. When in balance, support a well-being state of mind. Unbalanced can in particular reduce motivation, increase hyperactivity, reduce relaxation, and increase anxiety. Evidence connecting serotonin and dopamine to aggressive, stress-related behavior in dogs. In this paper, their research concluded a strong relationship between serotonin and dopamine in ADHD-like dogs, linking it with the aggressive activity and impulsivity. Decreased serotonin concentrations were also related to fear attachment problems and touch sensitivity. So what is it that affects serotonin and dopamine levels? Evidence connecting gut microbiome with levels of serotonin and dopamine. This paper identified that the influence of intestinal microbes could not be excluded in the release of neurotransmitters. Questions were left open on whether the regulation by gut microbes is direct or indirect. However, in conclusion, the studies reviewed indicated a close connection between intestinal symbionts and neurotransmitters in neuropsychiatric diseases via the gut-brain axis. So, is there a direct connection between microbiome and reactive behaviors? Evidence connecting gut microbiome levels with dogs displaying aggression and phobias. Research was completed on 3 groups of dogs, a group of 11 aggressive, 13 phobic, and 18 dogs displaying normal behavior, comparing the gut microbiome and the endocortical activity in the relevant groups of dogs. Their results illustrated what they described as a peculiar gut microbiome structure in dogs showing aggressive behavior, with high biodiversity and enrichment in generally subdominant bacteria genera. Whilst in the group of phobic dogs, they found the gut microbiome to be enriched in lactobacillus, a bacteria genus with no probiotic and psychobiotic properties. Recognizing the connection between the gut microbiome and the central nervous system, suggesting that it may be possible to affect different phenotypes in dogs using probiotic interventions to help restore a balanced host. With no defining evidence, we are left with the question as to whether diet caused the change in levels and resulting behavior, or if the levels changed as a result of the behavior. However, their conclusion was promising in the fact that they believed that the behaviors could be modified using prebiotic influences on the gut microbiome. So, can we affect this microbiome simply through diet? Evidence that nutrition can be used to affect microbiome levels in dogs. This study was specifically looking at the composition and development of the gut microbiome in young dogs, identifying how quickly it evolves during the early development of the dog and how the composition of the gut is affected by age, physiology, pathology, environment, medication, and nutrition, having either beneficial or harmful effects depending on the nature of the changes. Discoveries included that from puppy to adulthood, there is a natural and standard evolution of the core bacteria, and that control of these levels could be affected and altered through changes in the diet. Tests were done on increasing dietary fiber by adding beet pulps to diet and by increasing animal protein by placing dogs on a high raw meat diet. In adults, the microbial shifts were shown to reverse when the dogs were introduced back to their previous diets. With proof that microbiome can be affected directly by diet, it would suggest that the resulting behavioral neurotransmitters serotonin and dopamine would also be affected. Would it be possible then to identify the exact dietary changes needed to affect the correct microbiome that would consequently affect the specific neurotransmitters to cause a planned subsequent change in behavior? Evidence of alterations in diet affecting reactivity and aggression levels in dogs. In this study, their objective was to evaluate the effect of high and low protein diets with or without L-tryptophan supplementation on the behavior of dogs with dominance aggression, territorial aggression, and hyperactivity. When the groups of dogs were analysed, a lower territorial aggression score was obtained for dogs fed high tryptophan diets when fed a low protein diet. Dogs fed the high protein diet without tryptophan supplementation showed a higher dominance aggression compared with the others. Tryptophan is an essential amino acid that is responsible for the absorption of serotonin in the brain. It was used in supplement form during this research, but I would like to know if it would be possible to alter these levels on a whole food diet where a natural balance could be achieved. As sources of tryptophan are found naturally in cheese, chicken, egg whites, fish, milk, sunflower seeds, peanuts, and pumpkin seeds, which are all good sources of protein, it would suggest that it would be a challenge to naturally reduce levels of protein whilst raising levels of tryptophan at the same time in a whole food diet. So are there any other papers that discover a way of altering serotonin levels in the brain through a change in a whole food diet? Evidence of alterations in a whole food diet affecting absorption of serotonin by the brain. Scientists have found meals rich in carbohydrates promote an increase in insulin production, allowing muscle cells to absorb amino acids, making it easier for tryptophan to cross the blood-brain barrier, increasing serotonin levels in the brain. The research lacks any investigations into the effects of high carbohydrate diet, any negative changes in behavior following the increase of glycemic levels in the blood, along with any developing digestive and immunity issues. Is there an alternative way of affecting the microbiome diet whilst feeding a species-appropriate diet? Evidence that supplements can help to reduce fear and stress in dogs. This paper studied the effectiveness of a new dietary supplement derived from fish hydrolysate and melon juice concentrate, rich in superoxide dismutase, in reducing fear and stress-related behaviors in pet dogs. The trial results suggested that the supplement contributed to the observed behavioral improvements and could therefore be useful for reducing mild fear and stress reactions in the context of everyday life stressors, enhancing learning processes, promoting curiosity and familiarity with humans, and could be used in behavioral therapies. This paper researched one of many supplements that have become available to assist in the management of behavioral issues, anxieties, and reactivity in dogs. These supplements are now available on the market. Examples of supplements available on the markets to owners. There are many companies marketing behavioral changing supplements to add to your dog's diet. Although it is encouraging to see that supplements to a diet can affect a dog's relaxed state of being and reduce anxieties and reactivity, why would a diet require these supplements? If a dog's natural state is being calm, and this can be achieved by adding supplements, what is the dog's diet lacking in the first place? Is it unbalanced, nutritionally incomplete, or a species inappropriate diet actually causing harm? Evidence that supplements repair damage caused by inappropriate diets. This paper states that pet supplements for animals, as for humans, is to augment a diet lacking nutrient density to meet animal needs. This could be for maintenance, growth period, pregnancy or lactation, in rehabilitation or recovery from surgery. This paper suggests that if an animal was given a balanced and nutritionally complete species-appropriate diet, that there would be no need for supplements in addition to diet, suggesting that a correct diet has everything a dog should need to obtain a normal state of being. This would mean that the primary use of supplements is to repair damage that has been caused by an inappropriate diet. This would then support the theory that by feeding a species-appropriate diet, owners can have a direct impact on the effective microbiome levels and the balanced state of being of their dog, avoiding or reducing anxious and stressed states of mind, resulting in reactivity and aggression. Evidence that highly processed dog foods are species-inappropriate diets. This paper identifies a connection between ultra-processed foods in numerous chronic inflammatory diseases such as inflammatory bowel disease and metabolic syndrome in humans, describing the physical associations between the consumption of ultra-processed foods and chronic medical conditions, especially those involving the gut microbiome. It would be acceptable to assume that the same principle would apply to the canine world, given that we both share a mammalian digestive system, concluding that ultra-processed foods are species-inappropriate diets. And yet the pet foods that we find for sale in our trusted pet stores and veterinary surgeries are ranges of highly processed foods. So what would happen if we transitioned from a highly processed food, say kibble or canned, onto a mildly cooked or maybe a raw real food recipe? How would that affect our dog's microbiome? Evidence that dry processed dog food has a negative impact on gut microbiome. In this 4-week study, owners reported either improvement or no change in the health of the dogs transitioning from kibble to a mildly cooked diet. The diet change, however, led to a significant shift in the gut microbiome profile, and the magnitude of shift was associated with both the initial baseline of the gut microbiome and the change in the dietary protein. Evidence that dry processed dog food has a negative impact on skin microbiome. The gut and the skin are the two major defense systems for the body. It is interesting to discover through this research that the microbiome on the skin can be affected by diet,
and that this paper concluded that changing from a fresh food diet to a dry promoted a decrease in skin microbiome in dogs. Skin hydration was not recognized as a possible reason for this, given the moisture of the fresh food. Evidence that slightly cooked and raw diets have a positive effect on microbiome. The objective of this study was to determine the macronutrient digestibility effect on the gastrointestinal tract, fecal characteristics, urinalysis, and voluntary physical activity levels of the dogs fed commercial diets differing in processed types. The diets assessed were extruded dry kibble, high moisture cooked refrigerated, high moisture grain-free cooked refrigerated, and raw diet. In conclusion, the lightly cooked and raw diets tested were highly palatable, highly digestible, reduced blood triglycerides, maintained fecal quality and serum chemistry, and modified the fecal microbiome community of healthy adult dogs. These three sets of results suggest that a highly processed diet has negative effects on microbiome, and that a lightly cooked, fresh, or raw diet supports good gut health and microbiome levels, which in turn promote balanced levels of serotonin and dopamine. Evidence supporting the carnivorous diet of the pet dog. This paper addresses the fact that, though the dietary classification of dogs is omnivore, the origin of these omnivorous traits has been left unexplained. Data on the wolves' feeding ecology show that these ancestors of our modern-day dogs were adaptive, true carnivores and not omnivores. That wolves needed to be adaptive for survival during times of feast and famine and cope with a variable nutrient intake requiring an adaptable metabolism, which is still functional in our modern-day dogs. It identified that the nutritional characteristics of commercial foods differ in several aspects from a wolf's dietary nutrient profile, and this may pose physiological and metabolic challenges that dogs will need to cope with. Dogs have developed some additional production of amylase in their saliva and pancreas, explaining how dogs' digestive systems have evolved from the carnivorous wolf towards that of an omnivore, however not totally, and a highly omnivorous diet could cause negative effects in the long term, resulting in imbalance in the digestive and immune system, histamine, dopamine, and serotonin levels, affecting physical, mental, and emotional health. This would suggest that feeding a dog an omnivorous diet, which the processed pet foods in the market provide, would cause increasing cases of irritable displacement, anxieties, phobias, reactivity, and aggression.
Evidence that the pet food companies have done no or little research into the area of affected microbiome. This paper addressed the fact that within the pet food industry, a clear understanding of the impact of food choices on the microbiome and the overall health of the pet is lacking research. Identifying that pet foods are formulated to contain the typical nutritional building blocks of carbohydrates, proteins, and fats, with some understanding that there has become a need to add microbiome targeted ingredients such as prebiotics and probiotics. Each macronutrient can affect the composition and or the function of microbiome. Evidence suggests that the pet food dietary components may impact not only gastrointestinal disease, but also allergies, oral health, weight management, diabetes, and kidney disease through changes in the gastrointestinal microbiome. Until recently, their focus of microbiome research was to characterize alterations in microbiome composition in disease states, whilst missing vital research devoted to understanding how the pet food nutrition can change microbiome function and subsequently influence the animal's health. Their paper went on to say, pet owners have a role in shaping the microbiome of cats and dogs through the food they choose to provide. However, the pet food industry drives highly processed foods, with wordsmith marketeers using terms and phrases to suggest that their food is as good or even better than real and whole food. Pet owners are led to believe what they see and read, even if it is wrong, through misinterpretation. The pet food industry has flooded the market with a species inappropriate food, with a lack of knowledge of the damage they are creating, with marketing that misleads the public. These highly processed foods are disrupting gastrointestinal microbiome, affecting serotonin and dopamine levels, and causing irritability, frustration, reactivity, and ultimately aggression in our pet dogs. Summary of our literature review. We looked at how serotonin and dopamine levels in the brain affect relaxation and reactivity in dogs. How different types of gut microbiome affect the release of serotonin and dopamine in the brain. We recognized a direct connection with gut microbiome and reactivity and aggression in dogs. We discovered how different diets affect different microbiome in the gut. We discovered how highly processed diets negatively affect microbiome, serotonin, and the resulting behaviors, and that highly processed foods were actually species inappropriate diets. We discovered microbiome changing supplements could be added to processed diet to affect serotonin and dopamine levels and resulting behavior, and that supplements were needed when a species appropriate diet was lacking. We discovered that dogs' digestive systems are more closely related to their carnivorous cousin, the wolf, than the omnivores that they are classed as, and how a balanced species-appropriate diet positively affects the serotonin and dopamine in the brain and the resulting behavior. We discovered that the pet food industry has done little or no research into the damage their species inappropriate foods are doing to microbiome, and ultimately that the processed foods being sold to feed our dogs contribute to reactivity and aggression in dogs.